Hello and welcome, dear learners, professionals, and students, to the interesting topic on dissolution. That is, dissolution testing, the standard conditions and acceptance criteria for oral solid formulations containing highly soluble drug substances. The topic name is little bit lengthy, but the video will be very interesting. So whenever we are developing a dissolution method, any time you have thought that is there any standard condition to test the formulation, and is there any standard acceptance criteria, so that there will be a limited need for development of a dissolution method and giving a acceptance criteria. So the answer to this question is yes, there is a standard condition and acceptance criteria for dissolution testing, and this condition and acceptance criteria has some eligibility criteria. It is mainly done or used for oral solid formulations containing highly soluble drug substances. Mainly the oral solid formulation should be there, and it should contain highly soluble drug. Highly soluble drug, if you go by BCS, the soluble drugs are BCS 1 and 3. So there are different formulations for oral solid formulations like immediate release, modified release, and delayed release. Out of this, only immediate release formulation can be taken for the standard dissolution testing and acceptance criteria. The BCS classes are BCS 1, 2, 3 and 4, and out of these 4 BCS classes, the BCS 1 and 3 are those BCS classes in which the drug is highly soluble. Then if we think that why the immediate release formulation, solid oral formulation and highly soluble drug classes are selected for this criteria. See, because there is less risk associated with these formulations. That's why the standard acceptance criteria is given and standard dissolution testing conditions are provided. See, if you go to the basics, then you are knowing that drug absorption depends on the dissolution and drug solubilization in the physiological conditions, like the person has taken tablet by oral route with a glass of water per say 250 ml, and the formulation is IR type formulation, that is immediate release. It may be capsule or tablet, and that tablet or capsule get disintegrated and release the drug into the GI fluids. There the drug solubilization happens and the drug starts permeating through the GI membrane to the blood circulation. The permeation occurs after solubilization and drug goes to the blood circulation for systemic action. That's why dissolution is very important performance test which affects bioavailability, that is rate and extent of the absorption. In vitro dissolution data is a part of drug product application along with the CMC data, that is chemistry, manufacturing and control data. Then coming to the importance of standard dissolution conditions and acceptance criteria. As I have mentioned earlier that if you use this method and acceptance criteria, your dissolution method development will be faster. There will be, and if your formulation passing the eligibility criteria, then you can avoid the regulatory queries on dissolution testing. Also, the standard conditions and the acceptance criteria is considered as with a good discriminatory power, so that additional need to prove the discriminatory power of the dissolution method will be decreased, or the additional requirement of discriminatory power demonstration will be very less. That's why there is a much importance of standard dissolution conditions and acceptance criteria. If the product is eligible and passes this criteria, then you will have a benefit of faster dissolution method development, less regulatory queries, and there will be a less or no need of showing or demonstrating the discriminatory power. Then what are the factors affecting bioavailability? Solubility of the drug substance, permeability of the drug substance, dissolution of the dosage form, and pharmacokinetics of the drug substance. These are important factors and are required to be considered for dissolution development and making of the acceptance criteria, or setting the acceptance criteria for dissolution. IR drug products containing highly soluble drugs are considered to have low risk. These formulations are considered to have low risk in the bio studies. Impact on bio, that is bio availability and equivalence. And if the formulation meets the requirement of standard dissolution testing conditions and acceptance criteria, then we can say that, or it is generally understood that, the formulation has less risk on the failure of bioavailability and bioequivalence. That's why these standard conditions are made, and in detail we are going to study it. IR formulations which are containing the highly soluble drug substances are considered for this type of criteria and dissolution method. But if your formulation is ODT or sublingual, then this criteria cannot be taken into consideration, because for ODTs and sublingual formulations, the site of absorption is the buccal mucosa or oral cavity and not the GI membrane or not the gastrointestinal tract. That's why these formulations cannot be considered, and ODT and sublingual tablets or buccal formulations are not eligible for this type of standard dissolution conditions and acceptance criteria. Then those formulations which are IR but contains the narrow therapeutic drugs, they are not eligible. These formulations, though they are IR formulations, they cannot be applicable. If the IR formulation is containing a highly soluble drug substance, but it contains a drug substance which is used in emergency medication, that is also not a eligible candidate for this type of standard dissolution conditions and the acceptance criteria. Emergency conditions like COPD is there and rescue medications are there. So this type of medications, if are present in the formulation, then it is not eligible. Then the drug substance should be highly soluble. Solubility is a much more important criteria here, and the drug substance should be highly soluble across the pH range. That pH range is 1 pH to 6.8 pH, and the volume will be 250 ml. So solubility criteria becomes the highest drug product strength soluble in 250 ml or less of the aqueous media over the pH range of 1 to 6.8 at 37 degrees Celsius with a variation of plus minus 1 degree Celsius. So this is the solubility requirement. And one more criteria is there, that is highest drug product strength. If your formulation is having 3 strengths, like 100 mg, 200 mg and 300 mg, then 300 mg dose should be soluble in less than or equal to 250 ml of the media. If the highest strength is not the dose strength, like if 300 mg tablet is given as 2 tablets at a time, then your highest dose becomes 600 mg. And please consider this dose is not a maximum daily dose. This is a dose at a time only. So if 300 mg tablet is given as 2 tablets at a time, that means it becomes 600 mg. And that 600 mg should be eligible for highly soluble drug substances. Then another criteria is drug substance should be stable in GIT fluids. GIT fluids are 0.1 normal HCl and the simulated, that means simulated gastric fluid and simulated intestinal fluid. So there should not be any degradation in the GIT fluids. Generally, a degradation of less than 10% or equal to 10% will indicate that the drug is stable in GIT. Then manufacturing process should allow rapid dissolution. Your manufacturing process should be such that it allows the rapid dissolution of the formulation. Then the formulation should contain the excipients which does not affect or change absorption or permeation of the drug substance through the GIT, and it should not change or affect on the GI transit time. Like if your formulation is containing a excipient like surfactant or any other material which can affect the absorption, permeation or GI transit time, then the formulation will not be eligible for this. Now coming to the standard dissolution conditions. The awaited slide is here. Standard dissolution condition. So if you are using a basket method, that is USP apparatus 1, stirring rate or stirring speed or basket speed will be 100 rpm. Media volume will be 500 ml and media will be 0.1 normal HCl in aqueous medium. It will not contain any surfactant and medium temperature will be 37 plus minus 0.5 degrees Celsius. Now coming to the paddle method, stirring rate 50 rpm, 500 ml of 0.1 normal HCl in aqueous medium and no surfactant. So here the volume of medium is same, type of dissolution medium is same, that is 0.1 normal HCl, volume is 500 ml, only the stirring speed is changing. Here 50 rpm for paddle, here 100 rpm for basket. So this is the standard condition. If your product is capsule, for example, you have to go with 100 rpm basket. Why? Because capsule generally floats at the dissolution medium and it may give variation in the dissolution. That's why basket can be used, or pedal also can be used with the sinkers. So these are the standard conditions: basket 100 rpm, pedal 50 rpm, 500 ml of 0.1 normal HCl without surfactant, and temperature will be 37 degree. Then coming to the acceptance criteria, Q is equal to 80% in 30 minutes. So this quantity of label claim or percent label claim is 80% should be dissolved within 30 minutes. And always remember this Q is always given at stage S2. So example 1, here you have formulation with 98% drug release within 15 minutes and consider this as your bio batch. So you can give 80% dissolution Q point, and in S1 stage it will become 85, so that will also pass, and in S2 it will be 80. Then example 2, now in 15 minutes it is 75, 30 minutes it is 95. here in 30 minutes was 99, 15 minutes was passing. That's why 30 minutes can be given directly. But here it is 95% in 30 minutes. This is also passing the criteria. Now coming to the stage 3 example, it is showing 80% release in 30 minutes. So this type of formulation cannot be given a standard dissolution method criteria and the dissolution conditions. If you have given a dissolution condition which are standard, that is basket RPM will be 100, paddle will be 50. So something you have to do here to increase this dissolution, because if it is 80%, then as per the generalized criteria of biobatch minus 10% dissolution limit, it will be 70%. So let's go to the changes to the standard conditions. Can we change the standard conditions and criteria? So let's see what changes can be done and what changes cannot be done and should not be done. Can we increase the speed of paddle? Can we increase the volume of media? So yes, increase in speed of paddle and volume of 900 m can be done with suitable justification. Like if your product is showing coning at the lower speed, like 50 rpm paddle, then you can go for increase in the paddle speed. So directly we should not go to 75 rpm. In the increment of 5 or 10 we can go and we can provide the data. Like 50 rpm you have done, you go to 55, 60, 70, 75, like this. All the data should be generated. Sinkers can be used to immerse the formulation in dissolution bath. If your tablet or capsule are floating, spinning or dancing in the dissolution medium, that time sinkers can be used. Enzymes can be employed if cross-linking is justified with the formulations which contain gelatin, like gelatin coated tablet and gelatin capsules. Then other dissolution media than 0.1 normal SCL. Can we use the media other than 0.1 normal SCL? Because 0.1 normal SCL 500 ml is suggested, and if the requirement is that this media cannot be used, so other media can be used within the physiological pH range if you have a suitable justification. That dissolution media can be 4.5 pH acetate buffer or 6.8 phosphate buffer, or other media also can be used with the pH of physiological range. Then time point should not be changed. See, the 30-minute time point is fixed for this type of standard acceptance criteria, and Q point is also fixed and it cannot be changed and should not be changed. It is 80%. So in overall understanding, we should understand that the time point will be 30 minutes and Q point will be 80%. So less than 80% cannot be accepted. Time point cannot be stretched to 45. Then if you stretch it, it will not become the IR product containing highly soluble drug substances. If you go to 45 minutes, then you are hampering, or you are not, your product is not eligible for this criteria. So 80% is must, and this 30-minute time point is must. Then the main important stages are the dissolution method development, selection of the media, and all this paddle speed and basket speed. But if you use this standard dissolution acceptance criteria and standard dissolution testing conditions, then your dissolution method will be faster, you will have less regulatory queries, and the method of basket at 100 rpm and paddle at 50 rpm with 500 ml with the Q point of 30 minutes and release is 80%. It is itself discriminatory. So think on this topic, that why 500 ml is selected, why 50 rpm paddle and 100 rpm basket is selected, why 30-minute time point is selected. See, the 50 rpm of paddle and 100 rpm of basket is generally considered as discriminatory. So these are used. Then if the formulation is taken by the patient with 250 ml of water, then the 250 ml of media should be taken. But here 500 ml is taken, because 500 ml media is the generalized dissolution volume and it gives the less variations. And if you are using less than 400 ml media, then there are less possibility of dip of the basket into the media. Variations will be there. That's why 500 ml media is generally accepted. And if the dosage form is dissolving in 30 minutes at 80% level or above 80%, then it means that it is behaving like a solution and dissolution is not having much more impact on the bioavailability. That's why these conditions are derived. So I hope you might have got a good understanding of from this video on the dissolution testing for highly soluble drug substances, that is formulation containing highly soluble drug substances. So thanks for watching the video. Please do like, share and subscribe to this channel and press the bell icon so that you will get the notification of the new topics. Thanks for watching.